Welcome everybody. My name is Matthew Kaiser, and today I'm going to be talking about some gene panel validation workflows that are new in DNASTAR software. DNASTAR, as many of you may already know, is a company that produces bioinformatics software. We're located in Madison, Wisconsin. We have a nice picture of our Lake Monona here. DNASTAR has a long history in producing software at a very high level. Our developers, our sales force is mostly located here in Madison, Wisconsin. And we've been in business for quite some time. In fact, this year is our 30th anniversary, and we can see a nice picture here from 1984. We have our founder, Dr. Fred Blattner, who is still working actively with the company and coming up with great new ideas. And some of the folks here are some of our developers that still work with DNASTAR and develop new tools. So we have many, many years of experience producing the software, and on a regular basis, once or twice a year, we release new software to provide additional tools for our existing user base, as well as new tools for people that are new to the software. Our software, again, we can track it all the way back to 1985. It is research-grade software. It is the most cited bioinformatics software in peer-reviewed publications. You can see as recently as 2013, DNASTAR software is cited more than twice the next nearest competitor. So we've been in the business for a long time. We produce high-grade software, the wide user base worldwide. And it's really been our goal to produce software that people can use for research and publish the data that they get with our software. Excuse me. So with NextGen software, we can handle really any size of a project and a variety of different sequencing platforms. And so these are projects that can range in size from targeted gene panels to exomes to genomes, all the way up to multiple genomes. And so this can be Illumina or Ion Torrent or PacBio, Roche 454, or traditional Sanger methods. So the software has to be designed to be flexible enough to accommodate not only different platforms, but also different workflows. And so I'll show you some of the interface today on how we can set up projects. And today in particular, we're going to look at accuracy measurements and how we can verify accuracy when we're using different platforms and using different types of projects. So one of the first questions that will come up is, what is the power of desktop software? So, how can I use a laptop or a $2,000 desktop computer to handle these extremely large data sets? And so I like to show these benchmarks that we have for a variety of different data sets, from human genomes to exomes to cancer panels, AmpliSeq from Ion Torrent or TruSight from Illumina. And of course, these are different assembly types where there's different numbers of reads. So human genome may have 1.2 billion reads, as would 8 exomes that we align all together. And the coverages vary as well. So it's from 36x coverage in the human genome all the way up to tens of thousands of x coverage in a very deep cancer panel. And the assembly times for these large data sets vary anywhere from an overnight assembly, 12 to 13 hours, down to just minutes. So some of the smaller data sets like the cancer panel might be 10 or 20 or more minutes. And so the assembly in terms of speed is excellent. And I think that our software can outperform just about anything that's out there in terms of speed. Of course, when you're doing variant analysis, you also want to know not only is it fast, but is it also accurate alignment? And can we accurately detect SNPs or variants in these data sets? And that's really what the focus of the webinar today is. DNASTAR is providing researchers now a mechanism that they can assess the accuracy of their process, of their capture and downstream analysis. So that's kind of, so we're going to have new benchmarks then, in addition to speed benchmarks that can be based on accuracy. And so our SNP prediction accuracy with Illumina data is now greater than 99%. And this is really what we'll focus on here. And you can see the footnote here to the side is that this accuracy was determined using a human genome NA12878 reference material. And that's what we're going to dig into a little bit deeper. You know, when we make a claim of accuracy that's near 100%, how do we do that? And is it possible? It's one thing for a software company like DNASTAR to claim the accuracy and then have a data set that a researcher might not be able to get their hands on, or using a mechanism that is statistically too complicated to calculate the accuracies. What we've done with our latest software, our version 12 software, is we've put these tools in the researcher's hands so that you can determine this accuracy on, it could be the same reference data set that we use, or it could be your own data set using on your own set of standards. So we put the flexibility in the software to allow you to do that. When you use DNASTAR software for next-gen sequence data, you also want to be cognizant of the hardware that you may need to run the different assemblies. And this is just a basic schematic here. It's meant to be simple. And what I'm showing is that the ideal configuration for alignments to a human genome, so this is exome or gene panel or whole genome data, is really it's a three hard drive configuration. It is your computer's hard drive, which will run the operating system and the assembly algorithm. And that can be a 4-core computer, 16 gigs of RAM, a pretty modest computer. And then there's a disk called a scratch disk, and that's what processes temporary files. And so that can vary in size. 1 terabyte you may need for gene panels or exomes. 1 terabyte is going to be enough. If you're doing whole genome analysis, then maybe more, maybe up to 4 terabytes of space. So that's a dedicated drive. And then, of course, storage for both the input and output data. If you're going to be trying out a demo of our software, and you need to want us to check your hardware to see, if you have sufficient hardware, that's something that you can do with a sales rep. We can give you an idea of the hardware requirement for your particular project. But it's pretty simple hardware. These days, laptops often come with 16 gigs of RAM, so we're regularly running gene panel data sets, on a laptop computer. Even exomes can be run on laptops. Our software, in addition to great performance, there's also support, and the support varies. There's over 100 videos on specific topics on our website. So if you go to the support section, you'll see, Ion Torrent or PacBio or Illumina specific workflows. And these are usually, you can see from the screenshots here, 5-minute long videos that will show in detail a particular workflow. And so this is a great source of learning what the software can do and how to use the software. We also have webinars that we host from DNASTAR on a regular basis. So this is about once a month, and these topics can range from NextGen. So I had done one on gene panel workflows a couple of months ago, to basic molecular biology. My colleague Aaron did a cloning and primer design back in July, and my colleague Farhan will have another one next week on Sanger sequence data alignment and editing. So if you're interested in different topics, you can also visit our website. And these are recorded, and you can watch them from the past 3 years now. So the software that we're going to be talking about today is the LaserGene Genomics Suite. This is a suite of software that includes both the assembly software, that's SeqMan NGen, and then the downstream analysis tools, which is primarily our SeqMan Pro and ArrayStar softwares. And so the Genomics Suite then, it's available in all different kind of licenses, flexible licensing options. There are other packages that DNASTAR offers that focus on things like structural biology and protein folding and phylogenetic type alignments, and then the basics, cloning and primer design. So our software, we really strive to cover both next-gen sequencing workflows as well as the basic molecular biology workflows. So today we're going to be talking about a validation workflow that is part of really it's typically used for exome and gene panel workflows. You might also use it in a genome resequencing workflow if you have a validated genome. And it's right now it's a subgroup of our gene panel workflow. And you can see that our software is designed to do a number of different projects. So if you are in a lab where you're doing multiple things, this is an ideal software that can cover things like de novo assemblies of transcriptome or genome data, RNA-seq, as well as the workflows that we're focusing on today. So one of the aspects of our software that makes it somewhat unique is that it can be used to analyze a lot of different type of experimental setups or experimental designs. And so you may have, an individual that you've sequenced, where it's just one individual and you want to compare it to a reference and look for interesting SNPs. Or you could have a paired analysis of normal and tumor or pedigree. You just have a handful of cohorts that you're comparing, all the way up to association studies. And in our software, we can handle these different levels of projects. Association studies, it may be that you have 100 exomes and half of them are control and half of them are cancer, and you're looking for genes that are unique to the cancer group. So we have analysis tools then that can scale up to very large projects. And with the assembly speed that we have, you can do large projects, in a reasonable amount of time. We'll have some new options as well for these larger. If you are someone that's doing association studies and you've got 100 exomes that you have to do, we have some other options that will be available later this year. Things like assembly up on a cloud, where you can use cloud resources to handle these really large data sets. So that's something to keep in mind. And so we can analyze these different sample sizes, and really one of the challenges then with any next-gen data set when you're looking for variant analysis is finding the signal amongst all the noise. And there can be noise from the sequencing instrument, it can be noise from the alignment, it can be noise from the filters that are applied. And there's also the background, just the normal variations that occur, the 4 million or so that are unique to each individual. And so it becomes a really big channel then, filtering out the things that you aren't interested in to find just those few variations that are related to disease. So you have to have a great degree of accuracy then. So you want to make sure that you can actually separate that signal from the noise. So accuracy really becomes more and more important with these kind of workflows. So one way to filter out variations that aren't interesting is the use of gene panels. And gene panels are used by all the different sequencing platforms, so Ion, Torrent, and Illumina, as well as custom panels. And what their intention is to focus on hotspot areas and focus on sets of genes that are known to be implicated with particular diseases. And so it scales down the project for you essentially, so to limit the sequencing just to these areas. So for example, Ion, Torrent, the AmpliSeq Comprehensive Cancer Panel, it's targeting all the exons in 409 genes. Whereas the Illumina TruSight Cancer Panel, a little bit different panel, it's 94 genes, and there's almost 300 SNPs in those genes that are known to give some predispositions to different cancers. And then of course we have custom panels. So you may be designing your own panel or embellishing the panels that are commercial and using the latest literature to find new potential genes that you want to add to your panel. So if you're going to be doing panel design work, our software also provides mechanisms to assess the accuracy of the entire process. Is your targeting efficient? And then if you are targeting efficiently, are you finding all the SNPs that should be in those targeted areas? And so that's really what the focus is today, is how to validate the process in a gene panel or targeted resequencing workflow. Now, some of the important, I listed some of the files that are important for this workflow. That includes the input data. The input data is usually a FASTQ file as a starting point, or it could be an unaligned BAM file if you're an Ion Torrent customer. There's also a bed file that specifies the targeted regions, and a VCF file that is really contains a lot of information, but essentially it's a list of the high confidence SNPs that are in your panel. Now, of course, the commercial vendors will supply these files with their data sets, so you can download them. But there's lots of folks that have custom panels. They may or may not have a bed file or a VCF file. You may just have the SNPs in a text format. In that case, some minor formatting of a text file can get it into a format that is compatible with our software. So I'm going to talk about the bed file and VCF in a little bit more detail here. So bed and manifest files. These are typically tab-delimited text files that contain just the list. So the first column is usually a chromosome, the chromosome number, and then the start and end position of each one of the targets. And they're produced by Ion Torrent and Illumina both make bed files. Illumina may also call this a manifest file for some of their kits. And SeqMan then uses the bed file as a filter. So the aligner isn't forced to align the sequence data in these targeted areas. The aligner aligns the sequence data where it best matches, and then you can use the bed file then to filter and say, for example, only show me the SNPs that are inside my targeted regions. And so in many cases, that may filter out 90% of the noise from an assembly, which may occur in more ambiguous alignment areas in the genome. And so the bed file is very, very important then to use as a filter to find what's interesting. We have a tutorial then that can show someone how to convert a text file into a bed file. And this is a pretty common, if you're working with this kind of data, you may very well have just a list of gene targets. And so it is possible to make a bed file from scratch if needed. The VCF files, these are becoming more and more ubiquitous, and there's many different sources, Illumina and Ion Torrent, of course, but then also 1000 Genomes Project, the Exome Variant Server. Other software programs can all produce variant call format files. And SeqMan NGen, our software, can import a VCF file, and it essentially uses it as a list of important positions or reference positions. And we can use that to supplement the dbSNP data. So our software can also read the 60 million SNPs or so from dbSNP plus a VCF file that you provide to it. And those are all treated as reference positions or reference SNP positions. VCF files also have additional information, annotation information. We can utilize that as well in our ArrayStar software. We can pull annotations in and then decorate individual SNPs. In a SNP table using the VCF file information. And of course, these can also be converted. So two very important, for this kind of a workflow, very important file types. In this validation workflow, the VCF files are providing a list of the known variants in a control data set. And so it becomes central to the validation process. So the question of how is accuracy determined in a gene panel? So I have a couple ways that this is done. The first way is how most customers right now are doing things, at least the customers that I've worked with. Usually, folks that have been in the business a while have a number of data sets that have been sequenced, usually through Sanger, where they know that there are some verified variations, in some genes of interest. And generally, what you'll do is just run a next-gen run on that same data set and see, with my Ion Torrent or my Illumina, do I find the SNPs that I know are there? Which is a great, of course, a very natural thing to do, and you want to do that to make sure that as you move from Sanger to next-gen, that you're catching the same SNPs. And this works okay on smaller scales, but the problem with this approach is that usually there's only a few verified SNPs. So you might have a handful of genes and just a handful of SNPs. And it's not a large enough sample size to really get a feel for whether or not it's accurate or not. And generally, you're doing manual calculations, so you're getting a true positive detection rate and not really a good feel for false positive detection rates and some other statistics that go with it. So we think that a better approach is to utilize as large a data set as possible with your NGS data, and in something that has been sequenced with multiple sequence methods. So where there's millions of SNPs where you can get a true positive and false positive detection rate. And one of the ideal data sets for this is the NIST's Genome in a Bottle data set. And this is a human genome that's been sequenced with multiple methods, and we'll talk about that a little bit here. So the validation control is used to determine the effectiveness of the gene panel targeting. So this includes how specific are the primers, how efficient was the sequencing instrument, what is the accuracy of the assembler and the SNP caller, and in addition to the SNPs that are called, or the filters that are applied. And we can use the DNASTAR software then to validate the entire process. And so this is where the control, like the NIST Genome in a Bottle, is ideal. There are other control data sets. You may get one from Torrent or Illumina. They may have a control, sometimes even using the NA12878 genome. So you probably want to use both and see, do you catch all the SNPs that you should in a particular data set. So the Genome in a Bottle, the author of Genome in a Bottle is Justin Zook. This is human genome NA12878 Central European, sequenced by all different platforms to high depth. And what Genome in a Bottle is then is I have kind of this crude diagram. The green line is human chromosome 1, and in human chromosome 1 there are some regions that are difficult to get accurate data. And so the NIST bed file, which is the red line, shows the regions on human chromosome 1, and this is an approximate drawing, that can be called, or SNPs can be called at a high degree of confidence. So it's 90% or so of human chromosome 1. So these are regions where SNP calls can be made at a high degree of confidence. And so Genome in a Bottle then provides the bed file, so you know what the targeted areas are, and then provides all the high confidence SNP calls in these areas. Our software will use this bed file to limit these SNP calculations to these high confidence regions. Now, if you have a gene panel, which is much, much smaller, it may just have some coverage over the NIST bed file. We can make an intersection, that's the red rectangle, and actually make a smaller list of VCF variations where there's overlap between the gene panel and the high confidence calls in Genome in a Bottle. So we can use those then to determine the accuracy to find SNPs that are supposed to be there with this particular genome. So we've incorporated, because this is, there's a number of steps here that can be fairly challenging for a researcher to come up with on their own. We've incorporated this validation workflow in our gene panel workflows that was offered with version 12 software earlier this year. So our new gene panel workflows include a Mendelian or germline panel workflow, as well as a cancer or somatic workflow. And then, of course, the validation process is essentially its own workflow. And that's what I'll show you, is how easy it is to set that particular kind of a assembly up and then do the downstream analysis. So SeqMan engine is the starting point. We use it to assemble the FASTQ files. SeqMan engine is a 64-bit program. It's the one that requires the scratch disk. So this is, if you need to run assemblies, you usually want to put SeqMan engine on your sturdiest computer. And it has an interface then that allows us to set up these assemblies. And that's a key difference between SeqMan engine and the typical competitors for SeqMan engine, which are oftentimes open source programs that may not have a user interface at all. Excuse me. So SeqMan engine is designed then to be as easy to use as is possible, to be flexible, to allow for different platforms and different workflows, and reproducible. We can rerun a script if needed to load and rerun the exact same assembly that we ran in a previous session. And so I'll just show you some of the screenshots for what SeqMan engine looks like, and then some of the downstream analysis. So the starting point for SeqMan engine is usually a new assembly project, and we click next on the wizard, and that just brings us to the next page in the wizard, where we can choose the project type. And you can see the list here is growing. If you're familiar with SeqMan engine already, this list keeps getting longer as we find more project types. Of course, there's genome and exome. And with version 12, we differentiate between a Mendelian germline panel assembly or a cancer somatic gene panel assembly. And when you start to make selections in the workflow, then it'll start to set different optimal parameters for that particular workflow. So in a germline panel assembly, we're going to be expecting SNPs in heterozygous or homozygous frequencies, and so we apply slightly different filters. So the project, the workflow hopefully allows us to set up all different types of projects without having to answer too many questions that are difficult to answer in the wizard. So we choose the project type, then the assembly type, and again, this is a new screen. If we don't have a control, or if I don't want to run a validation control, I can run just a normal workflow, just run a normal assembly. However, if I want to run a control or have a validation set, I can select the templated assemblies with a control, and then click Next. And now it's asking for the template file. So what do you want to align your data against for variant detection? And so DNASTAR provides a number of genome template packages. A genome template package includes the NCBI annotated chromosome sequences. Those are in GenBank format. And the genome template package also includes VCF files, or may include VCF files, but it'll include dbSNP database files, Cosmic Cancer Database. You can add a VCF file, and that'll actually be in the next screen. At this point, we can add a manifest file, so we're aligning to the whole genome template, but we're only interested in the targeted area. So you can see at the bottom of the screen, I've loaded a targeted regions file, and it can be a bed file, it can be a manifest file, and that just tells the assembler that assemble the whole genome, but we're interested in these sub-regions of the genome. The assembly options now are default for the workflow that we've selected. So if we pick Illumina data, there will be a slightly different MER size and optimal match percentage, for example, than there is with ion Torrent data. And so, so the wizard fills in some of the optimal parameters for the end user. However, the end user still has control over things like minimum match percentage. And there's some advanced options here that I won't describe in detail that allow you to tweak different parts of the alignment algorithm. Now, prior to the validation controls, changing things from a default carried quite a bit of risk with it. DNASTAR has run hundreds of assemblies, with these default parameters, and really determined that for that type of data, that's probably the best parameter to use. Customers will often find that if there's a missed SNP, that they want to change parameters. So that may be done without a validation. If you change, for instance, a match percentage to find one SNP in a data set, you might actually be missing or becoming less accurate to optimize for one particular variation in a data set. It's kind of a dangerous thing to do. However, with the validation control, you can change these parameters and then get a statistical measure across a very large data set to learn, did you become more or less accurate for all the data points in that project. So it's a really nice, the validation really allows us then to make some changes from the defaults to see, did we get more or less accurate with the particular data set that we're working with. So it's a great way to fine tune and optimize by using one of these validated controls. So one of the so once the assembly is run, there's a number of reports that are generated, and the reports that are generated give different bits of information. There's some overlap, but different bits of information depending on what it is that you're interested in. So one of the first reports that's useful is a gene panel targeting report. And what this is telling me is if I've targeted 100 genes or the exons in 100 genes, did I get sequence data aligned to all of those targets? And if I didn't, can I go and investigate why I may not have covered a certain area? So this is an interactive table, and it is a report then that lists all the targeted areas. So what we have in the first column is a contig number, and then we have the feature name, and then the location within the feature. So this is going to be an exome coordinate. The length, so you can see these are all exon sizes. The coverage depth, so we have the depth of sequence coverage across that target. And then the percent of the target that's covered, and then an RPKM value, which is a normalized value, and then a read count as well. And so we can look at this particular table and then sort it. And for instance, in this particular table, I sorted by percent covered. And so I could see that in this particular panel that I've gotten coverage on almost everything, except there's 2 exons here where the percent coverage is less than 100%. And so when I'm designing a gene panel, and maybe I'm adding a couple more exons, or there's a new gene and I want to embellish it, so I design primers or probes to capture these regions. This report can quickly tell me, did I capture all the regions that I intended to capture? So you're not going into it blindly, or you don't have to search each exon. You get a report, and you can see from the 2 that I highlighted here that the coverage depth, that we have less than 100, but the coverage depth is pretty low for these, only 3 or 20x. So it's much lower than the other captured areas. So it's possible that for this particular region, the capture efficiency may have been very low. I didn't include a screenshot here, but there's also, you can see at the top of this table, there are tabs for targeted and not targeted. Because SeqMan NGen is aligning the sequence data to the entire genome, we can also go look at not targeted and see, did sequence data align to areas outside my targeted area? So for example, did I actually capture pseudogene and sequence pseudogenes rather than my genes? And is the data actually aligning to pseudogenes better than my target? So you can use this report then to figure out, first of all, did I hit all the targets? And if I didn't hit all the targets, figure out why all the targets aren't covered. So it's a really powerful tool for panels. So this is just another view then in SeqMan. I know there's a lot going on here, but it's you can see the report, the targeted report with the RB1 gene, just a targeted region selected. SeqMan Pro has multiple interactive views. So we're looking at a strategy view, which is the middle view. And that strategy view shows a depth of coverage histogram. And you can see the Y scale there is there's a 5,000 X deep. And I have it selected the same region in all 3 views. And so we can see then some green arrows there. Let's see if I have maybe a little highlighter tool here. So these little green arrows down here are exons. They're annotations on the chromosome sequence. And so maybe I'll try an arrow here instead. Yeah, that's better. So there we have some exons, and you can see that there's a depth of coverage then well over 5,000x over one of the exons. So in this particular gene, we didn't target all the exons, just a particular exon. And if you double click on this report, we also get an alignment view. So you can actually go and look at the individual aligned bases in detail. So it's a great way to go then and verify all the targeting, improve it. If you see that targets are missed, you can go back and redesign some primers and try to increase the effectiveness of the targeting. So that's one way to actually increase the accuracy, is to improve the accuracy of the targeting itself. So when a validated standard is used, another report that's generated is the SNP-calling accuracy report. And the accuracy report is generated from the standard that was used. So in this case, if it's the NIST NA12878 standard, and you have a targeted region, our software then can generate an automatic statistical report. And this goes back to the accuracy claim that we have of 99.8%. And this report will be generated in our ArrayStar software. So you run the assembly, say run validation report, and I just copied out a couple pieces of this, but you can see there's the first statement here is essentially a summary statement, and it's just saying that, given a minimum depth of 10x coverage, that we're detecting 15,494 out of 15,545 SNPs. using a filter, a probability filter of 75%. And so we can get a feel for, how many SNPs were there in our validated data set. So we have a little over 15,000, and we're getting most of them. But what does that mean exactly in terms of accuracy and how many false positives there were? That's what can be more difficult to calculate by hand. And so our report automatically generates this information. So it'll say the number of true positives detected was 15,494. There were 622 false positives. And from that information, we can calculate things like false discovery rate, which gives you an idea of how many false positives there are. And then we also get sensitivity and specificity. And from sensitivity and specificity, we can get something called balanced accuracy, which is just a simple calculation then. And you can see then from our data set that we can calculate accuracy on the genome in a bottle data set across a large number of variations. And that's really what's key, is we can then go back and change some parameters for alignment. We'll see, does this number go up or down? Not optimizing for one SNP in a data set, but really looking across 15,000 SNPs. Of course, this is a 15,000 SNPs, so this is in a exome analysis. And we are also working to generate some of the accuracy reports on whole genome and get all the statistics calculated for whole genome accuracy. So in areas that may not be as optimized as exome capture, but in some of the more difficult to assemble areas that are found in the genome. So one of the last things that you can do then, when you run the assembly in our software and get a targeted report and then a SNP validation report for accuracy, our software also provides a means to look at the assembled data, utilize a SNP report, and then visualize the aligned data as well. So you can do a visual confirmation. This is particularly important if there's something that is missed. So if you've got, for example, a list of 100 SNPs in your data set and you run a validation control and you find that one of the 100 is not detected and there's coverage there, those are the kind of SNPs that you want to have the ability then to manually investigate, look at the alignment, and determine what might be the cause. And there's lots of different, why isn't the accuracy 100%? So that last 1% or so, there's all different categories in that last 1%, and those are things that you'll investigate. It could be that there's an error in the reference that was used. That will show up occasionally. It might be that the sequence data itself isn't generated cleanly in an area like that. It can also be that the aligner, the SeqMan engine aligner for that particular SNP was not an optimal alignment, so the SNP could be missed that way. Or it could be a filter that was applied that just filtered out at a threshold. So there's lots of different reasons, but you need to be able to look at the assembly to be able to determine why a SNP was missed. And so, of course, internally we use this to make our software better. We've got this huge validated data set. We can keep tweaking parameters and trying to get to 100%, looking forward. For end users, this is a great tool then to find out how accurate is their process. Are they getting 100%, or if it's not 99.8, if it's something less than that, you'll be able to figure out why. And in the screenshot that I have here, you can see the SeqMan SNP report that is completely interactive with the alignment views. And the way the SNP report is structured, and I'll just kind of go through a couple of the columns here, but there's reference positions, there's a reference base and a called base, the impact. So there's impact on the protein. We had the SNP percent, and then whether or not it's in a region capture. By default, we filter. If you provide a bed file, there's a default filter applied to the SNP report that says only show the SNPs in the capture regions. And so again, this report has 11 SNP positions in it. So if I could turn that filter off and see how much data is there outside the capture. There's also a DB SNP ID. So this is all the DB SNP information that is has an RS number. So you can see that most of the SNPs I have in the SNP table have been identified and are in DB SNP. There are some in this column that do not have DB SNP identifiers. These are potentially novel SNPs to that sample. So we can apply different filters to focus on novel or known SNPs. And then we have a cosmic cancer database with its own identifier, the evolutionary rate profile information, the GERP, and then the user ID way over here on the right side. These are the actual SNPs in the VCF file that was used to validate the process. So these are SNPs provided, of known verified SNPs in this case that I want to see if we can detect them in a particular data set. So you can use this really nice SNP report then to filter through the variations. I can double click on any one of these SNPs and actually see the aligned data in SeqMan Pro. So we can see in this screenshot of an alignment, I have this top sequence here, the NC, it's human chromosome 4, and you can see the gene annotation and all the different mRNA and CDS annotations. And then down below, we've got the aligned data. And what I have highlighted there is a G to A SNP that occurs at 99.1%. It's NDD SNP. And it's a little more difficult to see highlighted, but the A's here are actually blue in color. So if you haven't used SeqMan software at all, it's top-notch software for looking at assemblies, large assemblies to human chromosomes, navigating through the assembly and looking at the actual aligned data to make validation. So again, it's a great new workflow that we have in DNASTAR software. If you haven't tried us out, I highly encourage that you try the new gene panel workflows. And with that, that's all I have today. If you have any questions, I will stick around here at the end of the webinar and answer any questions that you have. Thank you.